Good afternoon and welcome to today's panel discussion on triple negative breast cancer. My name is Nikki Kinloch. I am the CEO of Cure Cancer, and it's a pleasure to have not only our cohort of 2024 researchers here, but a lot of our donors, fundraisers and other supporters on the call today. Before I talk through the panel, as you see here, just a bit of housekeeping. So the call is being recorded for everyone's knowledge, and we will share this Zoom call next week for those that have missed out, or if you want to forward this to anyone, you can do so. And we also encourage conversation too. So if you do have any questions you'd like to either ask Nicole or Chen, please pop the questions in the chat box, which you can find at the bottom of your screen, and I will weave those into the conversation today. So what are we doing here today? Well, we're honoured to have two very special guests with us. Firstly, we have Nicole Freeman, a cancer survivor whose journey has inspired many of us here at Cure Cancer. In fact, we probably call her a friend by now. Nicole's first-hand experience with triple negative breast cancer has fueled her commitment to fundraising and advocacy, making a tangible difference in raising awareness about cancer research. And joining Nicole is Dr. Chen Li. Chen's a dedicated researcher focusing on triple negative breast cancer. This type of cancer is sadly a very aggressive form of the disease that lacks 3 common receptors found in other breast cancer types, making it more challenging to treat. This subtype accounts for 10 to 20% of all breast cancer cases, affecting a significant number of individuals worldwide. Current treatment options are sadly limited. Chemotherapy is the standard treatment for TNBC, but it is not always effective and can cause significant side effects. Chen's research is focusing on understanding the role of a particular protein in the development and the progression of triple negative breast cancer that is known to promote the growth and survival of cancer cells and is often overactive in TNBC. Chen is investigating how this protein activates specific pathways with the goal of identifying new targets for treatment, which can then help develop more effective and less toxic treatments for individuals. So please, let's give a warm welcome to both Nicole Freeman and Dr. Chen Li. Thank you both for being here.
Thank you.
Thank you. Absolute privilege and a pleasure. Thank you.
Now, we know you both well, but for those of you on the panel and also in the audience that really aren't aware of who you are and how you came into the Cure Cancer sphere, let's just talk about, rather than the triple negative breast cancer, but really your journey with us. So Nicole, do you want to kick off and start talking about how you came across Cure Cancer and why you are part of our family?
Yeah, absolutely. And I'm glad you're friends. Of course we're friends, you know, we're all here to help and do more. And I think it's incredible what Cure Cancer do, not just for breast cancer, but all cancers, which is, you know, really drew my heart in. But I was 32, diagnosed with breast cancer. This is my 10 years since diagnosis, so a pretty incredible milestone and achievement. But where I found Cure Cancer in particular was part of my, I'll call it kind of health recovery journey. Once I got through all the treatment bits and not just actually going through treatment, but the post-treatment process, because that is also a process that the patient goes through. I decided I had to get my body in fighting fit condition should this come back again, because, you know, as many on the call may know, and particularly Chen, you will know, triple negative breast cancer isn't always a one-off thing. There is a high risk that it could come back again within 5 years or appear somewhere else. So my mind went to get yourself in the best fighting condition, and that meant my health and fitness journey. And through that, started running with my husband and was literally doing Sedita surf and saw this beautiful colourful flag called Cure Cancer, and it drew my eye in. And the more research I did personally about what you guys did, I thought it just resonated with me. And I think there was things that I could do that might be able to help tap in and elevate my platform and just, yeah, get to know you guys and spread the word a little bit more, because there is a lot of incredible charities out there, but what you do really spoke to me because it's about helping the patients have a better quality of life throughout their journey.
Lovely. Thank you, Nicole. And Dr. Chen Li, you're one of our alumni from 2023.
Yep.
So can you talk to us about why you applied for one of the Cure Cancer grants to start with?
Yeah, so a little bit about myself first. So basically, I finished my PhD in 2016 at Monash University, and then I started my postdoc career, and I also went to Switzerland for another 2 years of training. So my lab, I have to give you some little bit background on my lab. So my lab is actually very, very famous in antigen processing and presentation, especially in cancers and and other autoimmune disease, for example. So that's why actually I think cancer should be my major career, major research directions, because as you all know, that is kind of really fatal disease, and again, it's kill a lot of persons, kill a lot of people every year. So our lab actually is targeting a lot of cancers. So TNBC is one of them, and it's probably most fatal one, actually, among the cancers we are actually investigating at the moment. So we have a lot of good research foundations, research basics about triple negative breast cancers. We published several papers about that, and this is actually came to my mind that I need to do something for these, because actually it's very kind of fatal, and it can lead to very serious consequences. So I think with the antigen processing precision state-of-the-art technologies, we can definitely do something just to improve the treatment, improve the antigen-based immunotherapy, just to be honest, to help the patients as much as we can. So this is how we actually came to this project, came to this specific cancer. Yeah, so basically a little bit about myself. So basically I'm a mathematician, so who actually used to sit in front of the computer for a whole day, which can be super boring. But with this project, I can do some experiments, which is fantastic. And I learned how to combine my skill sets of the computational stuff and the experimental stuff, which is quite important in my career development. And also I enjoy doing that because I think I'm helping people. At least I'm progressing my project, and maybe in one day we can have more effective immunotherapies for triple negative breast cancer.
Yeah, absolutely. Well, thank you both for that introduction. Before we jump in and talk to you, Nicole, Chen, I think it'd be good just to set the scene. In my introduction, I talked about how triple negative breast cancer is different. But do you mind going over that again to the rest of the audience? Conscious some people may not understand the difference between breast cancer and triple negative, and also why it is so difficult to treat as well.
So basically, triple negative breast cancer, as you have introduced, is actually accounts for 10 to 15% of the whole of the all diagnosis breast cancer cases. So the reason that we said it's actually very fatal or very serious cancer is because triple negative means that we have three receptors negative, three proteins negative. So we can't actually diagnose this or can't test it positive, these three proteins in these patients. So that's why we call this triple negative. So this kind of the main treatment, as you have introduced for triple negative breast cancer, is the chemotherapy and the radiotherapy. But as we all know, that is kind of triggered a lot of problems, because the patients usually have very serious side effects, and also they can develop a resistance to these therapies. And after 5 years, basically the reoccurrence of this cancer is quite high. A lot of people just unfortunately have that back. So compared to other breast cancers, this is very serious in triple negative breast cancers, and actually the 5 years survival rate or 10-year survival rate is quite low actually in triple negative breast cancer.
Thank you. So Nicole, back to your journey, if you don't mind just starting at the beginning of this. How did you know there was something wrong? Could you just talk to the audience about the discovery phase that you went through?
So work at pretty high stress, high corporate role, and running 1,000,000 miles an hour. So I was really run down, but for me, that was kind of life, right? Not a lot of sleep, run 1,000,000 miles an hour, go, go, go. that was probably the only thing when I did look back that was a bit of a marker that I probably should have paid a little bit more attention to. Just thinking, oh, I'm extra rundown and I just don't seem to be getting anything any better. It doesn't matter what I sleep, how much water I drink, what I eat. Then I found a lump in my left breast, and that was a Saturday afternoon, shared with my best friend and my husband at the time that night. They hit panic. I was like, oh, don't stress. Didn't really think too much of it. Went to the GP on Monday. Then I had a mammogram Wednesday, biopsy that same day, ultrasound that same day. And I went, oh, okay, this is picking up pace. Something's not right here. And within 7 days, I was in chemo, being told, brace yourself when this may not go in a fantastic direction. So get ready, saddle in, let's go, because triple negative, particularly even 10 years ago, the treatment was so aggressive and not a lot known. So it was kind of a let's whack you with everything and see how it goes in terms of chemotherapy. But I ended up having 3 tumours and it had spread through my lymph nodes. And really the only thing I felt was a bit run down. So it's a bit of a lesson to everybody to check, and don't be embarrassed or afraid to check. If it's nothing, great. If it is, then you deal with it and need to get on top of it as quickly as possible.
Yeah, look, absolutely. And how do you deal with that? And I'm sure there'll be many people on this call that have had cancer diagnosis, as well as we've got a whole cohort of researchers here too. You know, understanding patients, how they feel, the journey, and patient advocacy is so important to research. You know, how do you translate that in your mind from going to just being run down to sadly having triple negative breast cancer?
Suddenly you're in a chemo environment going, oh my goodness, it's pace. I think, you know, people talk about your mental well-being, and I'm a real big champion for that, because you have to make a decision to stay positive and fight it, because your body is going to give up many times and it's about to have a battle. And for me, it was a process of my mind and my body not necessarily always connecting, where I was like, right, this is a process, just get to the next chemo. Okay, here's the next marker, get to the next one after that. Or, okay, got to get to this date because then that's surgery. So you can kind of mentally brace yourself with markers and milestones. But your body's not following that. Your body's going, I need to stop, or this is too much. Or I actually had 3 code blues in my treatment journey, which, you know, can be a bit of a wake-up call, you know. So I think the other thing for me is 2 parts. Have an incredible bubble around you. I'm so fortunate. I still get emotional. My husband and my family and my best friends just rallied around me in a way that supported me how I needed to, but also respected my privacy and to get away and let me do this at my pace at other times that I need to. But I would also say, don't be afraid to ask your medical team questions, because they are the gurus and their job is to keep you alive. So no question is a silly one. And I carried a notebook with me, and my husband and I wrote down every question in between treatments and checkups, like, what about this? Or will this happen? Or what about when my nails fall off? What do I do? You know, things like that. So I would encourage patients to do that, because you'll get chemo brain, you'll forget what's going on, and it gives you a sense of control and self when you have those questions answered from your medical team. You're not alone, lean on them.
Yes. No, very sage advice. Thank you, Nicole. It's hard to think of what to do in that circumstance, because, as you say, your mind's dealing with so much
going everywhere. Control the controllables that you can. Yeah,
absolutely. So just moving back to Chen, you talked about wanting to do something to expedite this research, given sadly that the little progress we have had in this area. And to Nicole's point, back then the treatment was pretty savage, and everything was thrown at you because that's all they had. So you talked about this word antigen. Could you explain to all of us that are not scientific what an antigen is and what you're looking at?
Okay, I'll try my best. We talk about antigens. So antigens is something, it can be a protein, can be a part of the protein that can trigger the immune response of our body, right? So for example, when you get infected by flu, for example, we're going to have a lot of symptoms. And to be honest, most of the symptoms are actually the result, consequence of our antigen activation, immune system activation. So our immune system is actually taking actions to get rid of these viruses. So similar with cancers, basically these antigens. So our focus on is actually the protein antigens, or we say peptide antigen, which is part of the protein. So basically, our body is full of cells, right? So these cells actually living close to each other, and these cells are actually always communicating to each other. Like our immune system is currently recruiting through our veins, through our blood, to every part of the body, right? But they need to know, this immune system, these immune cells, they need to know whether is everything okay with every cell in the body. So basically, antigen here is actually it's actually part of the proteins so basically it's proteins have been chopped up into small fragments. So these fragments can be presented to the cell surface. So when the immune cells actually recruiting around and interact with normal cells, right? So these peptides on the cell surface can serve as a really important signaling antigen to tell the immune system that everything is okay with me or something wrong with me. So we need to take some action, right? So this is what we call, in this context, we will call antigen, right? So this antigen, if successful, actually can be activated, can successfully communicate to the immune cells and trigger appropriate immune response by the T cells.
And why does the body not recognize or respond to those cancer cells currently?
So basically, I remember Kelly previously talking about the cold tumor. So it's something related to cold tumor. So basically, sometimes the cells, just think about the cells are hijacked, right, during this process of becoming cancerous. So basically, from outside, it stopped presenting useful antigens. Those antigens can actually warn the immune system that, okay, I'm becoming cancerous, take some actions on me, right? But most of the cases, in lots of cancers, these cells are not able to present that useful peptides, useful signal peptides to the immune system, which can lead to very, very late recognition of these cancerous cells. So when immune system finally be able to identify, okay, something wrong with the body, often it's very, very late already. So that's why for a lot of cancers, when we have a very serious symptoms, it's already at this very terminate stage. So that's one of the many reasons. There's a lot of other reasons, but one of these is actually the cell is not able to present any useful signaling peptides to warn the immune system when it becomes cancerous.
Okay, great. So that makes sense. So essentially, it's very late diagnosis because the body isn't told essentially that the cells are cancerous.
So that's why early discovery, like Nicole shared, is fundamentally important, because a lot of cancers, they can actually be not cured, but actually if effectively controlled if it's been diagnosed very, very early, at a very early stage.
That makes a lot of sense, and we hear that often. Early detection is so vitally important. So with your piece of work then, could you explain what you're doing with the signaling and how you're trying to put in place a better signal between the body and these cells, and what are the next steps of your research?
So, okay, I try to make that as easy as possible. A bit difficult. As I said, so basically, we're not targeting the signaling peptide present at the cell surface to warn the immune system that something wrong with me, you should take some actions. But in the cell, generating these peptides, generating these signaling small players is kind of really complicated. So we are actually targeting some protein machineries where like a lot of proteins working together to generate these peptides and present these peptides to the cell surface. We're actually looking at these machineries and how they behave, for example, in healthy cells, right? In TNBC cells. And also, what we can also do is to stimulate these cells with cytokines, which I've heard that a lot of people have been talking about during the research symposium, which is quite nice. So cytokines is kind of a bunch of proteins that is signaling proteins that can help control the inflammation in your body. So the cytokines basically can generate a local inflammatory environment. So remember, like when we, for example, affected by flu, for example, and viruses, bacteria, and you can see, for example, you feel, for example, if your skin is infected, you feel that it's hot, rash, all these symptoms. That is because the immune system is actually taking actions now, right? So one of the actions is to release a lot of cytokines. These cytokines actually can help signaling between the problematic cells and the immune system that something wrong. Okay, come here, just do something, right? So we want to stimulate our cells like TNBC cells and see whether under this cytokine stimulation, if we can present more peptides, signaling peptides that can present to the cell surface to warn the immune system. So we actually saw something interesting that under the stimulation, we saw lots of interesting peptides popping out, which previously didn't really pop out without the stimulation. But what we don't know is what's the protein machinery within the cells can contribute to this, because this kind of a protein machinery can be used as a next level biomarkers, macromolecule biomarkers to help see whether people are, for example, response to the cytokines treatment or not, or these peptides, how these peptides can be generated. Can we generate more peptides, interesting peptides, by changing these protein machineries that that can be used to warn the system, the immune system, to take some actions to these cancerous cells.
Thank you. And we always talk about how the immune system and particularly immunotherapy is really important. The work that Chen is doing is cutting edge. Nicole, your journey, and congratulations on your anniversary. That's wonderful. You know, your journey was slightly different because research has moved so quickly, thank goodness, in the last 10 years. But can you talked about your chemotherapy? Could you talk about your treatment plan? And also, if you don't mind sharing some of the impacts that treatment had on you as well.
Yeah, wow, where do you start with that list? So I went through chemotherapy called the Red Devil chemo. So that was a pretty aggressive type back then that really zaps your body fast. For anybody obviously on the call that understands what that is. So that went from June till November period. Then I had surgery. Initially, I was told I was going to have both breasts removed, but thankfully, because of the aggression of the chemo, my tumours had shrunk in size. So ended up having a lumpectomy and all the lymph nodes out of my armpit. So that was a good outcome. Again, you know, that keeps a positive mindset going, yep, okay, we're getting this. I feel like I'm getting it. Then I went through 6 weeks of every weekday radiotherapy. So that was draining as opposed to painful, because your body is starting to make signs of recovery out of the chemo. It's trying to get itself back to normal. But then there's this new treatment that's kind of zapping your energy day in, day out. So throughout, I tried to maintain as normal of a life as I possibly can, because that kept me going again with my milestones, just get to this day or get to this marker. But that was kind of a bit of that really drained me, the radio at the end of all of that treatment. Then I went through 18 months of hormone replacement therapy. That was more I don't know what it's like now, but back then it was almost like a bit of a ribbon tying up the package going, just in case, let's do everything to try and kill off anything that might potentially be there, or just give you that extra oomph of protection. I was supposed to be on tamoxifen and that HRT for 3 years. I, in consultation with my medical team, made a decision to take myself off it because I was struggling. It was doing a lot of things to my body. And I still just felt like I was a sick person. I was like, okay, enough now. And if I had a 1% better chance of survival, I would stay on it. That was almost my thing. That 1% is going to be a 1% better chance. If it wasn't really or negative, I just removed it because I wanted to get my body back in a normal condition. But I think when you're going through treatment, it's easy to get your head around the process. You understand what chemo is going to do. Your medical team are there to brace you for that, but it's the after. You know, I was 33 and I went through medically induced menopause. You know, like, I'm a really young woman. How do you deal with that? You know, my poor mum hadn't even gone through it yet. So who do I talk to about that sort of stuff? You know, it was the bone density problems that I had. It was, you know, just pain in the body. So it was, whilst I'm not in an initial treatment phase now, your body is still really taking a beating. And I think that's something that people don't realize. Cancer treatment isn't just the chemo, the surgery, the radio, or whatever the drugs are. It's life after that. It takes a while to resume.
Yeah, absolutely. And thank you for sharing that. It's a stark reminder. We talk about it often.
Yeah.
At Cure Cancer, but it goes on and on and on.
Yes.
And it's much longer. There's a longer tail than anyone might imagine as well on your personal well-being. Chen, we talked about the start of your research and what you're trying to do from a communications perspective. What's the outcome that you are looking for from a research perspective? Where are you hoping that this analysis, and we'll talk about your data sets soon, but where are you hoping this analysis will take you in your research?
So I would like to see the very important thing that we want to see is actually what protein machineries plays the most important role in presenting these diversified stimulated peptides, these antigens to the cell surface and contributing to the the immune system response. Because these protein machineries can be used as a biomarker for us to see whether patients are going to respond to this or not, how well it's going to respond to this. So that's kind of a really important question that we want to answer at the moment.
So would it be fair to say that the hope for the future with this research is that we can almost offer personalized treatment to patients based on how their body is responding to certain treatment plans?
This is actually a very important question because now people are talking about generic treatment like chemotherapies and everything. But I think I'm sure that Nicole, for example, you probably have a lot of these conversations with your medical team about this, because I recently have a chat with a medical team from the Alfred Health, so on a different type of cancer. So which we talk about this like, so they are actually the immunotherapy team, right? So they told me that, Chen, you know what? So 50 to 60% people, right, just don't respond to the immunotherapy at all. So the other 50 luckily respond to this. But there's no gray area. Either this patient can totally respond to this immunotherapy and get really, really positive results. But either this totally no response at all. So we actually targeted to both like generic and also personalized. Just we want to answer what in general, like what's the most prevalent effective drug can be used for a big population cohort. But on the other hand, it's also important to see personal medicine or the patient specific situation by doing our, for example, similar experiments here and see whether we can find some specific drug for them.
Makes sense. Thank you. Just going to pause there and welcome. We had a couple of new guests. We're talking to Dr. Chen Li and to Nicole Freeman about triple negative breast cancer. If you do have any questions, please pop them in the chat box. I do have one question. It's very scientific. So
not for me then.
No, not for me either. Maybe you can translate what this means as well, Chen, for those on the call that aren't scientists. But the question is about the cytokine-induced killer cell therapy in breast cancer and whether it's a reality now.
So to be honest, cytokine-based therapy is not well applied at the moment in the clinical settings. That's what I know, because I've been talking with all these medical teams about this cancer whatever cancer type that is, but in general, the immunotherapy. So most common one still like chemotherapies and the radiotherapies. But a lot of research, I have to say, is they have shed a light in this cytokine-induced killer cell therapy. But I don't know anything that has been widely applied, established in actual clinical oncology, medical oncology.
Lovely. Thank you for answering that. So just moving to your computational analysis. So you touched on it briefly. You said it's very boring. I think that's very unfair because you are making a difference. So when people talk about researchers or scientists, a lot of them think about those that are in a wet lab with a gown and some gloves and a telescope. Now, you're not one of those. So can you explain the difference between what the wet lab people do and what you do?
Yeah, this is a little bit funny to talk about every time I explain this to my friends. So yeah, so basically you know what's happening in the lab every day is actually we do cell culture, right? We do sequencing, we do lots of different analysis. So the result of this analysis is just a bunch of data. It's a pile of data. So basically the problem is nobody knows actually how to effectively dig into this data is kind of a big problem now, because all this experiment is done, but how to interpret useful results from this data is fundamentally important. So as a research mathematician, I think the job is to use whatever you can, programming, AI, machine learning model, whatever. You want to effectively dig into this data and get something useful. So that's what the routine job is for a research mathematician. But I can say that a bit different now because I also wear a gown and lab coats and you do some fancy experiments in the lab. So I can now understand both of the sides, which is quite fascinating.
Yeah, absolutely. And on that subject, I mean, we talk about that often, that patient advocates and patients that are going through treatment or have been through treatment are so critically important to researchers. And notwithstanding that most researchers are not clinicians, so they're not actually doing the treatment, they're in the labs, they're on their computer. I'll ask the same with Nicole in a second, but Chen, why is it so important to work with and liaise with patient advocates from your perspective?
What I have to say, I think everyone would agree with me, is it's not just a job, right? You can understand it as a job, like, okay, I come to my lab in 9 to 5 and then do my experiment and then I leave, right? But it's actually not like that. Behind that is everyone's life, is everyone's health. So that's kind of makes different from the normal job, like what you work in the office or somewhere else, right? So you are having this huge responsibility. So by interacting with these advocates, these consumers, you will have a very good understanding of how serious these cancers can impact people's life. How serious, because you won't be able to understand that until you have you have cancer or you have close friends who have cancer. Because to you, it's usually some stories on the TV, but you don't really understand what really means to people. So my experience interacting with advocates just make me feel super significantly like immersed into their life. Okay, so this is daily life you have. This is the pain you have. This is why everyone is expecting you to make a difference. So you will feel like you have a huge responsibility. So that's what I think is the most important thing for me, to better understand disease, to better understand the life, how their life was impacted.
Thank you. And we talk about this often. It's not just about trying to find a cure or a quicker diagnosis. It's actually about ensuring that the quality of life whilst you're going through the treatment is bearable as well. Nicole, from your perspective, what systems and processes or communications did you find supportive whilst you were going through your treatment?
Well, first, I love that you said quality of life, because I think that's the beautiful part about this whole cancer community with your medical team as well as patients or past patients is it is a community and everyone really is rallying around you. And I think, you know, for Chen and I want for people on the call, know that we do appreciate that. When you're in the hot seat and you're going through it, everything you tell us and everything you guide us with is so critically important, and it means something to us. But I think for me, it's about asking lots of questions. It's about doing your research. It's about that notebook I talked about with everything. And every single time you go and meet your medical team, ask them, what else should I be doing? Who else should I be speaking to? In your experience, what else do you think I should do? Or if this were, I remember asking a question of my oncologist, if this was your sister or your mum, would you be giving them that advice? And, you know, just really lean into that sort of power. But also, companies like yourself, like with Cure Cancer, why it's important to me is to spread that word. And we say awareness, and awareness doesn't just mean raise money to save lives for the research. It's also to raise awareness for people to reach out and ask questions. Through some of the cool stuff we've done the last few weeks, Nikki, I've had 2 girlfriends reach out to me that unfortunately have been diagnosed with breast cancer. I had no idea, but they hadn't shared their journey yet or their story because it's just been overwhelming for them. So when I say awareness, it's also just letting people know you're out there and letting them reach into you to ask those questions that you might be embarrassed about or nervous about, or it's overwhelming or it's too hard. And not everyone is as fortunate as I was in a position of, I had financial comfort that I could go and get the best treatment. I live in a metro city. I could go to a nearby hospital. I didn't have to travel a couple 100 kilometers down the road for a basic treatment. And I had a beautiful family support system and best friends that rallied around me. So again, everybody has that. So that's when I hear this word awareness, it means so many different things to me.
Yeah, absolutely. And I mean, this is why we do what we do at Cure Cancer. It really is.
The platform. Yeah.
It's about funding early career cancer researchers. It's about funding innovation and kickstarting new ideas. But at the end of the day, we're doing that because it will result in a change in what's happening to positively affect patients, which is so important as well. And that's something we all need to remember as we work through research projects and the funding. We are creating change and we are making a difference, and some researchers going a lot quicker than others, but even now talking to Chen about his research, you know, it's having an impact on what is about 20% of all breast cancers. So that's, you know, a significant impact as well.
Massive.
Chen, you were a Cure Cancer grant recipient last year. Could you share with us your journey since you've received the funding and how important it is to receive early career research funding?
Involved a lot of head scratching when I was trying to put in my application, and this is a lot of work actually, but the journey has been really rewarding, I have to say. This involves all this, for example, design and writing and the actual experiments, and also the summarizing everything. So I can't exactly remember how I got to know Cure Cancer. I think I got Cure Cancer from your website. I think this is a long time ago, but I think my experience with Cure Cancer has been really pleasant, I have to say, because I think Cure Cancer is really great platform for early career research, especially for early career cancer researchers, which is quite important now in current very competitive research culture, I have to say. And also been involved in a lot of like events like fundraising and some other just interviews and everything. I think it's really nice. It's kind of it promotes actually our research a lot. So it's really, really nice experience with Cure Cancer.
Thank you. And we talked about obviously your research on triple negative breast cancer. You're also researching in other areas too, but collaborations are incredibly important as well, because as we like to remind ourselves that it's not just treating cancer patients in Australia, it goes on to be able to treat patients globally as well. Can you just talk a bit about that impact from your research and what that might have to other patients around the world?
So basically, we in our research now, so when we design the research, so we are actually targeting, so we basically think about different type of populations that can be affected. So basically, we're now actually targeting, for example, different HLA types. So the HLA types is actually a human antigen types that can vary a lot across different populations. So we definitely include as many as we can, so that our results can be specific to different HLA types, which means different human populations from different ethnic groups. So that's what we actually are working on at the moment, and definitely we keep this going in the near future. So that's, I think that's that's current plan of our plan to cover as much as we can, cover as big population as we can.
So are you saying that the cancers are different within different ethnic populations?
No, it's basically, so remember we said this antigen is presented to the cell surface actually by what's called HLA, so human lymphocyte antigen, right? So this antigen varies a lot across different individuals and has different preferences in different groups, ethical groups of people. For example, in Caucasian, we have some mainly predominant HLA types. And for Asian people, we have another predominant HLA types. So we're trying to cover as many as we can. And then this is basically our strategic plan to address this problem and see whether we can make our results more compatible to different ethical groups.
It's fascinating. Thank you. Nicole, from your perspective, the advancements in research, have they affected you in your cancer journey? Have you seen an impact over the last 10 years?
I've actually seen impact through friends that unfortunately had to now go through the breast cancer journey. One of them did have triple negative, which I kind of went, oh wow, I've got a friend that's going through it. But the advancements I've seen are incredible, and just little things like I lost my hair obviously very quickly through chemotherapy, and that identified me as a sick person to the world, even though I was trying to hide and go through my positive mindset and go through those stages of just getting this done. And I wasn't ready to be seen as a sick person. Whereas now there's cold caps and there's different technology. You can have cold gloves to protect your nails, because losing your nails is incredibly painful. People probably don't think about that, but my gosh, it really is. Everything you touch now hurts. So it's those sort of little things in your daily life that impact your recovery journey and the impact that it has on you. But I've definitely seen all sorts of progression. The number of doses of chemotherapy appears, from what I understand, to have reduced, and some of the severity has reduced. So change is happening. It definitely is. And every time I go in for my scan, I don't necessarily always have to have a PET scan, which is the weirdest thing in the world when you're being pumped through, with that dye, and gives you that warm, strange feeling before you have your scans. And now I don't need to. There's other technologies. So I think it's reassuring to me to know that the efforts that researchers are doing and the funds being raised are actually going into life-changing and life-saving and life-improving technology.
Yeah, absolutely. Thank you. And Chen, from your perspective, where do you hope and how quickly, it's always the question, million-dollar question, how quickly do you think that you'll be able to see, or are you start seeing results already from your research? Because everyone wants to know how long will it take to get from the clinic, from research into the clinic?
This is kind of a yes, definitely million-dollar questions, though very stressful questions, because usually from research to animal models and then to clinical settings, it usually take a long time, unfortunately. But we still we're still actively progressing our project in the lab at the moment. So this very hard to say the specific timeline for these, but we can only say that we are trying our best to accelerate this as fast as possible.
Absolutely. And what are your hopes, Chen, for the future with your research?
I think I would hope in my future research or current research progressing with the current research progressing, I would hope that we can develop both generic and personalized therapies for breast cancer, for triple negative breast cancer and other cancer types, because I realized that this is getting really this is a really big problem. Every time I talk about this with medical oncologists, so we need to hurry, and we need to make difference. We need to get. We are actually now combining all different cutting edge technologies to do personalized sequencing and sample specific personalized sequencing, and now we're just diving into the huge amount of data and trying to find the biomarkers, personalized biomarkers, and also the biomarkers that can be applicable to a huge population cohort. So that's my hope. Actually, I really hope that we can be successful in both ways, both directions.
Thank you, Chen. And Nicole, as we wrap up, I'll leave the last comments to you. We have a whole cohort of researchers here and also a lot of our supporters. What are your words to both the researchers and those that have supported Cure Cancer?
Oh, first, thank you. Complete and utter admiration. You know, having gone through it firsthand, what you do matters, and it really makes a difference in someone like my life. And you know, as the logo says, we all hope to cure cancer, but you know, my hope is that it's also reducing the severity of the treatment, because curing cancer, you know, completely is probably an unrealistic thing today, but reducing the severity and the impact on the patient would be my wish. And I also hope that every person just thinks about the platform they have. You don't necessarily have to work for a large corporate and tap into big fundraising efforts, but just think about what you can do, whether it's donating $1 or, you know, having a ladies at lunch that I used to do way back when I started my fundraising journey with a group of girlfriends and my family, just to give a couple of dollars and have a great time. But we can all just do that one little thing that makes such a big impact. And unfortunately, if the cancer diagnosis isn't a positive one, at least you help that person and the carers around them, their journey going through that. So yeah, but nothing but appreciation and admiration for my support network, but for people like yourself, Chen, for what you do, because I get to clock off at the end of the day and call it a day, but you take that home and it's a lot. So thank you.
Well,
It's
thank
my
you. I
pleasure.
couldn't say that better. Thank you to all of you here today. Nicole, thank you for sharing your
Thank
very
you.
personal journey. Chen, thank you to you and your team.
Thank you.
Incredible work. And to the other researchers and also to our donors and fundraisers, I think it's fair to say, in your words, Nicole, that what you do matters. Raising funds and donating is so important, but all of that money goes to funding incredible researchers like Chen and the rest of you here. So know that you are making a difference. We have seen several advancements, most recently in cancer research, and it's exciting, the progress we're making. And we couldn't do that without donors and fundraisers. So thank you all. I wish you a good afternoon, and we'll chat later.
Thanks, Nikki.
See you
Thanks,
later.
Chen.
Bye. Thank you.